The information covered in these notes is from chapter 14-1 and 14-2 of your textbook. The information that we're going to cover today is on karyotyping, and we actually discussed karyotyping briefly when we covered meiosis. And we did it in a context to introduce the words diploid, homologous pair, and haploid. But right now we're going to talk about essentially more detail about what exactly is a karyotype, how they're made, and information you can learn from looking at a karyotype. So to start, the study of chromosomes chromosomal alterations and related diseases is called cytogenetics. And cytogeneticists study an individual's chromosomes from the nucleus of a single somatic cell. And somatic means body cell, not gametes. So somatic cells are your skin cells, your muscle cells, your liver cells, your blood cells. They're not egg and sperm. Now, in order to look at these chromosomes, typically the cell is not going to have chromosomes formed because the cell more than likely is in interphase. So the scientist actually has to condense the chromosomes by adding chemicals. And then they remove the chromosomes from the cell. And then they are put into what you know as a karyotype. So in order to do that, the chromosomes first actually have to be put onto a slide. And that slide is going to look like this. So notice that this is not organized. The chromosomes from one cell are just smeared into a region that you can see under the microscope. The slide is then photographed. And now we have computers that can greatly speed up this process. The homologous pairs can then be put together. And I'm going to draw a quick example here. A non-replicated chromosome, remember, is going to have a centromere in the middle. And we want to put homologous pairs together to make our karyotype. So the scientist that is probably using a computer to help them is going to look for certain characteristics of the chromosomes in order to put the homologous pair together. And so that's called morphology, or the physical appearance of those chromosomes. Now, what they look for is homologous pairs are the same size. The centromere is in the same place. The top and the bottom sections of the chromosome are called arms. And so this kind of goes with letter A. The arm length is going to be the same for both the chromosomes in the pair. And then lastly, chromosomes themselves don't have any color unless they're stained. So they do have to be stained. And when they are, they have dark and light regions. And that creates what's called a staining pattern. And so homologous chromosomes are put together because their staining pattern is going to match as well. So size of the chromosome, oops, I missed an E, centromere position, arm length, and staining pattern are how the homologous pairs are put together. Now, if you're wondering what are these chromosomes that look like cartoons, that's exactly what they are. They are just a cartoon depiction of what the real chromosome, which is that, what they look like. And it turns out that genes are located in certain regions, and these regions have numbers. So for example, 15 is going to be, the region 0.3, 0.1. So you would learn about that if you ever took a higher level genetics class. But this was chromosome 5. And you can see the staining patterns here in the cartoon, the dark and the light regions. Okay, so homologous chromosomes are arranged on a karyotype from largest to smallest. So the largest chromosome is chromosome number 1, and the smallest chromosome is chromosome 22. And the sex chromosomes are going to go at the end, and I'll talk about those in just a minute. The short arm is oriented toward the top. So what I mean by that is if you have a chromosome that's centromere is not exactly in the middle, the short arm will go on top and the long arm would go on the bottom. They are also grouped A, B, C, D, E, F, G with the sex chromosomes at the end according to common physical features. Similar size, and again, centromere position. The final karyotype would look like this. So chromosomes 1 through 3 are group A, 4 and 5 are group B, 6 through 12 are group C, 13 through 15 are D, 16 through 18 E, F, G, with the sex chromosomes at the end. Now, karyotypes can tell us about chromosomal abnormalities. We can see any abnormal numbers of chromosomes, broken, missing chromosomes, chromosomes that have extra regions that shouldn't be there when a karyotype is done. Now, what would actually cause a person to have more or less chromosomes than they should have? Normally, a human has 46 chromosomes. And typically the answer is because there was a problem in meiosis. And that problem is called non-disjunction. And we learned the word disjunction before. It means to separate. And so when you have non-disjunction, it's a failure to separate. During meiosis 1 or 2. So in meiosis 1, remember we separate the homologous pairs. So if you had non-disjunction in meiosis 1, you would have a failure of separation in the homologous pair. In meiosis 2, you separate the sister chromatids. And if you had a failure for separation in meiosis 2, they wouldn't be separated. So either one could happen. And what would end up happening is it would result in a gamete with 2 copies of a particular chromosome, and other gametes would not have any copies of that chromosome. So essentially, chromosome number is affected. in the gametes. If you'd recall, we want to have 23 chromosomes in the egg or the sperm, so that when they join together, in the zygote is going to have 46. If that number is incorrect due to non-disjunction, you will end up with the zygote either having more or less chromosomes. If non-disjunction happens in meiosis 1, all the gametes will be affected. If non-disjunction happens in meiosis 2, half of the gametes will be normal and half will be affected. And that is described down below. Here's meiosis 1. Here's where non-disjunction happens in meiosis 1. You can kind of have a dividing line. Here is where the non-disjunction is happening in meiosis 2. If it happens in meiosis 1, the homologous pair doesn't separate, and then you end up with 2 gametes that have 1 too many, and you have 2 gametes that have 1 too few. But all are affected. If you have non-disjunction happening in meiosis 2, well, half of your gametes are still going to come out normal. So you're going to have 2 gametes that are normal and 2 gametes, that are affected. 1 gamete is going to have 1 too many. 1 gamete's going to have 1 too few. Now, what can happen if a gamete that has experienced non-disjunction actually is the gamete that is involved in fertilization? Well, you can have a offspring that has a genetic abnormality, and they are typically called trisomies or monosomies. Trisomies mean the organism has 3 copies of a chromosome when they should only have 2. And monosomies mean they have 1 copy instead of having 2 copies. You might wonder, well, what about more than 3? What would happen then? And the answer is it can happen only in the sex chromosomes, but any other chromosome is typically fatal. In fact, there's only some chromosomes that can even allow for trisomy and the offspring still survive. Most trisomies are fatal. So I'm going to talk about the ones that are survivable. And I'm going to start with autosomal trisomies. So these are the ones that are happening that are not involving the sex chromosomes. So there's 3 copies of an autosome, and they are trisomy 18, trisomy 21, and trisomy 13. Trisomy 18 is called Edwards syndrome, and there is some intellectual disability, pretty severe, defects in the hands and the head, eyes, and the ears. Some of the other phenotypes are there is a clench with an overlap of the fingers, often malformed ears, very prominent heels in the baby. And this disorder in particular, a lot of children do not live past infancy just because it's so severe. Now, Down syndrome is probably the most survivable and common trisomy, the autosomal trisomy. It's very survivable. These children are often able to eventually support themselves, have a job, live on their own. There are some phenotypic differences, a stocky body type, sometimes some speech defects due to a larger tongue. There is intellectual disabilities with some individuals. But this, again, is trisomy 21, and this is a very small chromosome. So it's possible that because this chromosome has fewer genes on it, it is more survivable. Finally, the last trisomy is Patau's syndrome, which is trisomy 13. These children will often have a cleft palate, polydactyly, which means they have an extra finger on their hands, malformations of eyes and ears, a smaller head, also intellectual disability. And again, these children possibly are not living very long lives because of their disability. Now, with regards to sex chromosome trisomies, and I'm going to get to monosomies in a minute, these are going to involve the sex chromosomes. So this is the 23rd pair. They are not autosomes. And sex chromosomes influence the genetic sex, not the gender, because gender and sex are different. But the genetic sex of the organism is influenced by the chromosomes. So the first trisomy is called Klinefelter's, and that is XXY. Now, typically, genetic males just have a X and Y chromosome. So the extra X is the trisomy there. And these males are often taller than average, and typically they are sterile, meaning they cannot have children. But many actually do not even know that they have a trisomy. It also often does not present itself until puberty. Sometimes poorer hair growth, a tendency to lose chest hair, possible breast development, possible lack of a deepening of a voice in puberty. But other than that, those who have Klinefelters typically are leading totally normal lives. It does not affect their intellectual abilities, school, anything like that. And then there's male and female trisomies. So if it's a genetic male, XYY, and there's an extra Y. And if it's a genetic female, XXX, so there's an extra X. There was, I had read somewhere that there are some possible learning disabilities, but I've also read other articles that say that that is not true. So there's kind of conflicting thoughts out there. However, physically, no abnormalities, very few. And it is very possible to have a male or female trisomy and never know throughout your life. And then monosomies, the only survivable monosomy is where you have one copy of a chromosome, and the only survivable one is monosomy of the sex chromosomes for females. So girls who are X and no other chromosomes. So they're supposed to have two X chromosomes, but they only have one. This is called Turner's syndrome. And I actually had a student about maybe a decade ago who had Turner's syndrome. She was extremely open to talking about it. She was in a 3-level biology class, just like you. And she was pretty open about talking about how she couldn't have kids herself. She was a little bit shorter than average. She, again, could not have children. She had some bone issues. But other than that, she was leading a very normal life. But she was missing an X chromosome in all of her cells. So that is the only survivable monosoma. You can't have a autosome and only have one copy of it. It is fatal. So again, karyotypes from the top, how they are made, how you pair the homologous chromosomes up, what a karyotype looks like. Looking at a karyotype, you can see if someone has extra or missing chromosomes. Then the next part of that would be, well, how did the extra or missing chromosomes occur? Well, it occurred because of non-disjunction and meiosis. And then what are some examples of some disorders due to extra or missing chromosomes? And they're called trisomies or monosomies. And that is it.